Okay, so this is lecture 5, and in the last lecture, we looked at a microscopic technique where we could produce a true three-dimensional image, and this is called confocal microscopy. And the basic way it works is that with fluorescent microscopy, we have this excitation and emission. So we excite the fluorescent molecule, and then we detect the light that will emit. And utilizing something called a confocal microscope, we can actually measure the emission from a single plane. And so in this example, there's 9 different planes that have been scanned. And we call these Z-stacks. And if you put these together, what happens is it builds a true three-dimensional image of our object. And so we looked at a little video that showed the reconstruction of a small section of tissue where we were looking at a neural network. To get better resolution, we need to utilize electron microscopy because the electromagnetic waves have a much shorter wavelength than visible light. So visible light, we're limited to the shortest wavelength which is violet light, and that's 390 nanometers. And so utilizing electromagnetic waves, we get down to, I believe we said something like 0.004 nanometers. And so this allows for much improved resolution. Keep in mind, you can't do electron microscopy on living samples. So unlike all of the light microscopic techniques, which have the potential to be done on living samples, depending on what the sample is and how the preparation needs to go to visualize them, we don't have that option with electron microscopy. And if we look at transmission electron microscopy, it's basically the equivalent to bright field microscopy, in that we're shooting the waves through the sample and seeing where they're blocked. In order to do this, we need to introduce electron-dense dyes, and these are in the form usually of heavy metals, and so that will allow the image to be seen. And there's all kinds of different staining techniques, and there's different ways you can stain to look at different features, but we just sort of looked at a straight-up transmission electron micrograph. And so with this, you get resolution of sort of 1 to 2 nanometers. I should also point out that transmission electron microscopy has a low depth of field. So you can see you need to actually use a very narrow sample, and you can only envision a very narrow sample. Whereas for scanning electron microscopy, this has a big depth of field. So the example we looked at, the photographer or the microscopist had put a whole Drosophila fly into the system. The way this works is similar to the way our eyes work. Our eyes, we see the light that's reflected off of objects, so I can see all of you because the light is reflected towards me. In this case, however, it's not light waves, but instead these electromagnetic waves that are generated. And they have to bounce off something electron dense, so we start by just coating our entire sample in a very, very, very fine layer of gold. Okay? Just a couple of atoms thick. And so that will reflect the electromagnetic waves, and instead of collecting the image at the bottom, what we do is we collect the image at the sides, so we're catching the reflected rays. We talked about a couple of other things, but one of the last ones we looked at was this idea that eukaryotic cells are highly compartmentalized. And that increases the efficiency of processes by, first of all, allowing us to concentrate nutrients or substrate, whatever is required for that process, and also to limit negative interactions, so competing interactions with other processes. And the organelles in a eukaryote, many of them are completely enveloped or surrounded by either 1 or 2 lipid bilayers, so membranes. And we're going to spend, I think it's topic 3, looking at the organelles in some detail. We can see many of them in this particular picture here that's looking at a eukaryotic cell. So in today's lecture, we're going to talk a little bit about cell culture. We're going to look at properties of culture media, because that was a real breakthrough moment in cell biology when media was developed to support growth of mammalian cells. And we'll probably get, depending on the time, maybe a little bit into our section on biomolecules. So the part about biomolecules, the reason that we talk about it in cell biology is that we need to kind of understand the building blocks, the little things, before we can build up to the larger things. But again, it'll be a pretty brief overview. So for cell culture, really what we're talking about here is we're growing cells outside of the organism. So we can take a sample of human tissue and we can grow it in a cell culture outside of the human. Primary culture is taken directly from an organism So we take a, say, mouse liver, we break the cells apart, we put them into culture, and that's called a primary culture. If we do a subculturing, subcultured cells, what happens is we've taken a primary culture and we've grown those cells, and we take them out of that culture and put them into a new culture. And so most of the cell cultures that you would be dealing with would be secondary in nature. They're not coming directly from a tissue. Okay? We'll look at some examples, perhaps, of primary culture, but most of the ones we're talking about are what we would call secondary culture. These are terms that, on this slide, that the first and the last are ones, you're not going to see these that often, but I'm going to tell you what they are anyway. If we look at a mass culture, using that term mass culture, we're looking at a large number of cells. And it's kind of a generic term for a lot of cells, as you can probably tell by the name, mass culture. It doesn't give us a lot of details about the cells. It might be all from one organism, from multiple organisms. We don't know, according to the term. And so it's not a term really that you see very often. Clonal culture, this is more important because the cells all arise, the cells should all be genetically identical. They all arise from either a single cell or a colony that is from a single cell. So if you look at a clonal culture, they should be genetically identical. And so you're more often going to be seeing work with things like clonal cultures because often when we're working with something, we want all the cells to be the same, have the same genetic makeup. An axenic culture has a single species in it. And again, almost all the cultures that we are dealing with are axenic, and so it's simply a term that isn't used very often. Okay? I can't really think of any situation where I've seen them with respect to growing mammalian cultures for axenic. We talk about them sometimes if we're reconstituting organisms. We take all their bacteria away and we introduce a single species. That's where you might see that term, but that's not something that really is going to apply to our 225 crops. So the big term here is this clonal population, where they all arise from a single cell or a single colony. There's different ways we can grow culture, and so growing cells really is very important for the field of studying cell biology. this ability to grow cells in culture. So typical culture techniques would be either what we call suspended or plated. And if we're looking at suspended cultures, these are growing in a liquid media, and the cells are suspended in that media. So usually we have some sort of shaker or rotation or something to keep those cells suspended. Because of course, within a solution, cells are going to slowly settle to the bottom. And you're probably familiar with working with suspension cultures. You've maybe looked at bacteria before. We'll be working with yeast in this lab. And so many types of cells grow well in suspension culture. For mammalian cells, this is used less often. For reasons we'll get into in a few minutes, mammalian cells, many cell lines of mammalian cells are not particularly well suited to growing in suspension, which is unfortunate because, as I said, it's a cheap and easy way to grow cells. The cells could be, say, suspended in a nutrient medium. So you have some semi-solid media, kind of like a barely set jello, I guess would be the best way to describe that. And the cells grow inside that. You may be familiar with using a more firm, or what we would call a semi-solid surface. And this would be things like the agar plates that we work with in microbiology, or if you're plating yeast cells, you'll plate them on a semi-solid agar surface. And of course, we have Fanny Hess from Robert Cook's lab to thank for the, she was the first one to develop agar plates. And so these would, I imagine, be techniques that most of you are familiar with. At least agar plates and growing in suspension. And you've probably seen this, you've maybe even performed this, although I know many of us missed out on experiential experiences last year. For mammalian cells, it's a little bit different for reasons that we'll get into. And so very often to grow mammalian cells, we need to grow them on an actual solid surface, a solid substrate. And this is normally going to be plastic. And so we grow these on plastic or in plastic culture flasks or dishes. And those dishes, the surface of them for many types of cells has to be treated. There has to be something that allows the cells to stick. And the cells have to stick in order to survive. And so there's different ways we can treat the surface, but one of the ways that's quite interesting is that the surface is treated with components from what we call the extracellular matrix, which we'll come way back to much later in the term. The extracellular matrix. And the extracellular matrix is everything that surrounds the cells in your body. And a good example of a component in the extracellular matrix is something called collagen. I think it's collagen, like that. And so the surface of the plastic is coated with collagen, and that allows the mammalian cells to anchor there, so to grab on and hold onto the plate. We're going to talk about normal cells and cancer cells, which we can also group in with what are called transformed cells. And if we look at normal cells and you take that culture dish and you turn it on its side, the cells grow a single layer thick. Okay, so we call that a monolayer. In contrast, many cancer cells or transformed cells can actually pile up and form multiple layers. And that can be observed here, where we have this section here with multiple layers. And in this example, because it's just part of the plate that has this sort of clumped up, we can call that a foci. A foci is just an area where things are concentrated. Okay? Any questions so far? So have you, as a group, have you ever done any cell culture? So bacterial, yeast, anything? Just put up your hand. Yeah, so most of you have, but not everyone. You'll get lots of opportunity in the lab to work with this coming up starting this and next week. Yes?
Can you repeat what's a foci?
Sorry?
Can you repeat what is
Oh, what is a foci? A foci is just the term just means a specific or a small area, a concentrated area. And on this particular slide, shown in the red, there's a little clump of cells that are growing more than a monolayer high, so we can call that a foci. Because when you're growing the culture, they'll first show up as those foci, and you might get multiple on a plate, and then they'll continue to sort of grow up from those foci. Yeah. Okay. So we're moving now to talk about mammalian cells. Okay. And so animal cells are what we're mostly going to be interested in. Although this, yeah, so let's stick with the terminology. Here will be normal versus transformed. So normal cells are non-transformed, non-malignant. These are non-cancerous cells. And there's some interesting properties to cells when you take them out of the organism. And when you take them out of the organism, they're what we call anchorage dependent. So they have to be anchored in order to survive. And we'll actually describe the reasons for that much later in the course. We're going to circle back to this. But for now, we just need to know that if they lose their ability to hold on to the surface, the cells actually die. And so this is why we have to treat the surface of that dish with something that allows the cells to stick. They're dependent upon what are called growth factors. So growth factors are sort of a cocktail of signal molecules. And these signal molecules tell a cell, first of all, you have to keep telling a cell to stay alive, oddly enough. If you stop telling a cell to stay alive, its default is to destroy itself. They also contain signals for growth and proliferation. So the cells grow bigger and they divide. And you need to provide these signals to normal cells because their reproduction, their growth and replication is very highly regulated. And so we have to add in these factors to do that. And what we'll often do is provide serum. So what you do for serum is you take a sample of blood, and usually you use fetal blood for this because that's a phase of rapid growth, and also fetal blood is, of course, going to be sterile. And so you basically clot the blood, and the stuff that's left over, you give that to your cells. And so for cell culture, this is, of course, the big expense is the serum is expensive. But the other thing about the serum is that you don't really know what's in it. You have an idea, but because it can switch from batch to batch, this is something that we can call this undefined. Okay? If something's defined, we know everything in it, and this example is undefined. So you could do an analysis and figure out what's in it, but normally you wouldn't do that for an experiment. You know roughly what's in it, but the serum is this one thing, this one, and in many cases, only thing in the media where we don't know exactly what or how much of each thing is in it, okay? The cells will start to grow on the plate, but when they touch each other, they stop growing. And so we call this contact inhibition. When they come into contact with each other, they cease to grow, and this is why they form a monolayer. And the last thing we have here is something called the Hayflick limit. And the Hayflick limit is interesting in that cells will divide a certain number of times and then they'll just stop. So in culture, this happens in culture, cells will double maybe on the order of 50 to 100 times, and then they'll cease to replicate. So you'll get no more cells out of that culture. Yes?
How do you provide a serum and not know what's in it? So like, where do you get the serum from?
You just buy the serum. You buy it in big containers, and then you just add, we'll see the recipe for one set of media in a few minutes. So yeah, you just add it in, so yeah. So, got two more questions, one there, and then one a bit farther back, so go ahead. Sorry, I'm just going to move a bit closer. I don't have the best hearing. Sorry.
I'm saying if the serum is undefined, does that affect the defined variable?
So if it's undefined, does that mean it's variable? Sorry, was that the question?
Yeah, it's like you have two independent variables, right?
Sorry?
You have two independent variables, right?
Yeah, so, right. So because the serum is somewhat variable, now it's not hugely variable. This could mean that, yeah, if you're doing experiments, you have to be careful you're using the same serum for all of your samples, for example. And between experiments, you always need to compare with the same serum. And it actually can make a difference, amazingly. So, yeah, good point. Back there.
Sorry, why do they stop growing after they come into contact?
It's this thing called contact inhibition. The question was, why do they stop growing? And so what happens is that when they come into contact, it initiates signaling pathways that prevent growth. And this is one of the reasons that these are the sorts of measures that are in place to stop your cells, your tissues, for example, from massively overgrowing. So growth is always highly regulated. Yes?
If they stop growing when they come in contact, how come they make it foci?
Okay, so the question is, if they come into contact, why do they form foci? They don't. So if we look here, normal cells won't form any foci. So they'll form a monolayer, and we can see this in this slide here at the top. So yeah, good to reiterate that though. So they won't form, normal cells won't form foci. In contrast, transformed cells, and the same properties would be with cancer cells or malignant cells. The difference there is that, well, we can use those terms interchangeably. We're going to use those terms largely interchangeably, okay? Cancer, malignant, transformed cells, okay? So it's a totally different ballgame with these. They aren't anchorage dependent. They don't require serum. They don't have contact inhibition, and they are what we call immortal. They have no Hayflick limit. And so many of the cell lines that you'll work with, if you ever go on to work with cell lines, most of the cell lines that you'll work with in a laboratory situation will be these immortal cell lines. Okay? And so there's other differences with those two. We'll sort of bring those up as we're moving through the course. And again, we circle back to this whole section when we're talking about cancer. Okay? So this is the big difference between our transformed and non-transformed cells. This slide, we have time culture in days on the x-axis and the number of cells on the y-axis. And we're looking at transformed versus non-transformed cells in the presence or the absence of serum. And we can note that normal cells without serum experience very little growth. When you add serum in, though, we see that they do grow, and they grow to some limit, and of course this is once they formed a monolayer. We know that they stop. Monolayer. In contrast, when we look at the malignant or transformed cells, they grow with or without the serum. So this is an interesting note that I want to point out. And this depends on sort of your approach to your science. So who knows what in vitro and in vivo mean? Okay, you want to tell me what in vitro means? I caught you totally out there. Yeah. So basically, for in vitro, the term means in glass. And what this means is we've separated something from its system. So if we're looking at a human and we start studying glycolysis in a test tube, we call that in vitro. And in contrast, in vivo means in living. And so in vivo means you're within the living organism. So what is cell culture? Is that in vitro or is that in vivo? Who thinks it's in vitro? And who thinks it's in vivo? Most people thought in vitro. Some think in vivo. So in fact, you'll see cell culture described as both, depending on your perspective. So my initial training in life was as a biochemist, and a biochemist would tell you that cell culture is in vivo, because you're looking at an intact cell with all of the biochemical cellular pathways and processes. And so I would describe working with a cell culture as an in vivo experiment, because I'm used to actually just isolating the enzymes and not having any living component there. Then I moved to become a cell biologist, and then you talk to cell biologists, and they would call cell culture in vitro. Because for them, in vivo means working with, say, a mouse model or an intact organism. So that's just something for your interest, but it is something that you will see in papers where sometimes you'll see cell culture described as in vitro and other times as in vivo. And so there was no right or wrong answer to that last question. So, yeah, oh, sorry.
Can you go to the back? So if you want to see if the cell is cancer cell or not, can we take a biopsy and then put it in the lab?
Oh, and see, so could you use this?
Add the serum and see how much it grows?
Yeah, so the question is, could you use this to test for cancer? I guess technically you could. Not all cancer cells will actually establish as a culture. Cancer cells are actually, they're sick cells. They're not normal. Something we'll come back to really late in the course. So they won't necessarily grow in culture. And also it's a slow way to examine them. So it's a pretty slow way to look at them, and it would be quite expensive. So normally for biopsies, a lot of it's just visualization. So you use different stains and you simply do an analysis, but it's very fast. Yeah.
And if the cancer cells don't grow in culture, how did they come up with that?
Oh, some do grow in culture. I'm going to get to that in everybody's so excited about cell culture, but we'll get there in 4 slides, I promise. Yes?
Just quickly, do you get the serum by putting blood in a centrifuge, or is there different
You just clot it first, and then you can centrifuge it out. Yeah, I've never made serum, but that's the idea behind it. Yeah. Okay, so where are we? Oh yes, okay.
Sorry, a couple slides back, I was just wondering, on the normal cell slide, what HDX meant? Yeah, one before this, or one after this one. Yeah, down at the bottom.
Oh, that's human diploid fibroblast cells.
Okay.
Yeah, it's just a type of cell, so yeah, sorry about that. Yeah, it's just a particular type of cell. It's not really that important. So if you're trying to set up a primary culture, you start with a tissue of interest, and you have to break the cells apart. So you can do this enzymatically or mechanically. Often you might combine both. So you need to break the cells apart. And the cells are in a tissue are held together by protein interactions. And so those are the types of things like adding a protease. Proteases cut proteins. And so that helps to break the cells apart. You need to then put the cells into a suitable environment. So proper temperature, proper amount of carbon dioxide, and most importantly, a media. And that media will allow them to grow. Okay. And we can see a visualization of the steps in this slide here, where at the top we're starting with a blob of tissue. In this case, it's being homogenized, so you use, it's sort of like a mortar and pestle, and you smash up the cells, and you may combine that with a proteolytic treatment. The cells are then put into a primary culture. Once those cells grow, as can be seen in the second plate from the bottom, they're now a monolayer. These aren't going to grow anymore. You can take some of those cells off the plate. So you basically have to treat them so they come off of the plate, and then put them on a new plate, and now from this point forward, you've established a secondary culture. And unless you take steps to transform this into an immortalized culture, you can do this subculturing maybe like 50 to 100 times. And so for many purposes that you might be doing, this is all that you would need. You don't need necessarily an immortalized cell line. I should point out I've never worked with primary cells, so I don't know how difficult they are. I've worked with a lot of immortalized cell lines, and the techniques are fairly straightforward. Yes?
Why would you do a secondary culture? Like, why do you just not use
Oh, why would you do a secondary? Just because that's not very many cells. You can't really do an experiment on like a single plate of cells. So if you take them and split those cells into 4 plates, then you'll get 4 plates that are confluent, and then you can keep splitting those, so you can split them up to a point where you can do an experiment.
more samples, basically?
Yeah, you do have more cells, yeah, so that you can carry out a reasonable experiment. So that's why you would do these subculturing. Yes?
How do they know when to like stop, I guess? Because you said like 50 to 100 times you could do that. Like, how does it know?
Oh, they'll just die.
Oh.
Yeah. So someone's asking how you know when to stop. That's actually not up to you. They'll just die. Yeah. Yeah. This is looking at the first sort of commercially available successful media. All right? And so looking through here, this sort of covers into the next slide as well. So you can sort of work off whichever slide you want to. This is kind of like a summary of it, but I'm going to work from this one looking through the actual components. And so this was called Eagle's basal media. And the first thing that is provided is a number of amino acids. And what are amino acids for again? Right, so that's what we make proteins out of. And humans, so there's 20 different amino acids. And we'll look at this in our biomolecules section. And humans can make about half of those, and the rest we have to get from diet. Okay? Some organisms can make all of the amino acids. We're not able to do that. And so the ones that we can't make, we call essential. Those are the essential amino acids. Okay? The ones that we can't make. The ones that we need to get in our diet are called essential. Anything that we talk about as essential when we're looking at nutrients means they have to come from diet. So, yes?
What would be an organism that can do it, like with 20 amino acids?
Lots of bacteria can do it, yeah.
Is there any like major organism
No animals can do it, no. Yeah. We then have vitamins. Vitamins are also essential components, and we utilize these, enzymes utilize these to carry out their function. And again, these are things that can't be made, and so they're provided in diet. Again, there's organisms, lots of bacteria that can make all of this, but not animals. Salts, we require salts to maintain tonicity. So what you need to do is you need to balance the salts inside and outside of the cell. Otherwise the cell swells up and explodes or shrinks and dies. So tonicity, we need to make an isotonic solution, so the concentration of salts in and out are the same. And salts are also required for a number of cellular processes. We'll talk about salts, we'll come back to them a few times in the course. Yes?
So what does the cross means? Like the amino acids and amino acids, what does the cross mean?
It's just some footnote for the table. Yeah. I don't have the footnotes provided. Yes?
Essential means that we can't make it ourselves.
We can't make essentials,
We
no.
have to take it from our diet.
Yeah, right. Like essential oils, you have to buy them from a pyramid scheme. You can't make them. So you can just think of that as our example. But yeah, so essential amino acids it's about, I think, 9 that we can't make. And the other ones we don't make well, so we provide them in the diet.
Out of 29 of them, we cannot make.
It's about, well, it's like, it depends on what you're kind of reading up. So it's around 10, 11, we can't make those, and those are essential. And we can count up this list. How many are on here? 1, 2, 3, 4, 5, 6, 8, 9, 10, 11, 12, 13. So some must be made at low-ish amounts.
And if you weren't able to receive some of these in your diet, what would happen?
You would just die. So if you don't get these in your diet, you would die. Because you require all the amino acids. I know, it seems like a lot of tragic endings in today's course. Boy, what a downer. A few miscellaneous things that are included. Where's my pen gone here? So glucose, what do you think glucose is there for? It's food. This is energy. So it's required for energy. We also get carbon out of it. Penicillin. So we add penicillin, which is an antibiotic. I think you all know that. But the reason is that all these things, these nutrients, also make the media really, really a great place for bacteria to live. And our cells take around 24 hours to divide, but bacteria can divide in 20 minutes or so. And so if you get a little bit of bacterial contamination in there, and you go to look at your plate 2 days from now, it's only going to have bacteria because they're so fast at growing, they'll just outcompete the mammalian cells. So often there'll be different antibiotics, and this particular one has 2 antibiotics, penicillin and streptomycin. Okay, these function in different bacterial pathways, so it targets 2 separate pathways, the cell wall and translation. Phenol red is an indicator dye that goes from pink at pH 7 and it turns yellow as the solution acidifies. And so this is often added because as your cell culture uses up its media, the waste products are acidic, and as they start to build up, the media will turn yellow. And this is a good indicator because often you're running hundreds of these plates at a time, and you can quickly look and see which ones you need to replace the media in. And so it's a really fast way to know what stage your media is in. It's not always in there, but it's almost always in there. And so all of these, what we notice here is that for everything, and again, I'm having some issues with my pen, but for all of these, we know the exact amounts of everything. And they're added in these exact amounts. Except down here, we see there's serum. We know the amount of serum, but again, the exact components in that serum are undefined. Is there any questions about any of that? Yes?
So phenolite is just an indicator? It doesn't like neutralize the acid or anything?
No, it's just an indicator. It just tells you it's just a quick way to tell the pH of a solution. Some of you may have seen those in chemistry class. It's anything you can see quickly, visually make a determination is really useful. Yes?
So what is this basal medium that we just learned about it? Like all these things?
So this is a media for growing cells in. So the question is, what is the media? So the media is the liquid that we grow cells in.
But didn't we say it's undefined, the thing that we add to the culture?
The only component in this one that's undefined is the serum.
Oh,
We don't know what's in the serum necessarily. I mean, we know mostly what's in it, but you don't know how much of each growth factor is in there, for example.
serum is something different than the medium.
The serum is one component of the media. So we can see this tells us down in this corner, this tells us how much serum we put in. Yes?
I just didn't catch what streptomycin did.
Oh, streptomycin, the question is, what is streptomycin? That's another antibiotic. So we have two antibiotics in here that target different bacterial pathways. Yeah. So I promised I would get back to this, which is the first cell culture. It's the first cell line that was established. So researchers had been trying for quite a long time to establish cell culture. They were able to grow some cells for a short period of time, but there were no immortalized cell lines. The first immortalized cell line was developed at Johns Hopkins in 1951. And this was work done by George and Margaret Gay. And George was kind of the clinical end, and Margaret worked on the culturing end, developing media for allowing the cells to grow, because that turned out to be a big piece. And the first cell culture that was established, they tried a number of different samples from different cancers, as we were talking about earlier, but most of them wouldn't grow. And this lady, Henrietta Lacks, she had a particularly aggressive cervical cancer. And so a sample from this was taken, and it turned out that this was the missing piece. And this grew as an immortalized cell line. Now, of course, the story of this has probably become clear, and many of you may know about the story. There's a famous book, The Immortal Life of Henrietta Lacks. And the problem here was that this had been done without Henrietta Lacks's consent or permission. So in the United States, you may not know this, but there's no free medicine, and so many people can't afford medical care. If people require care, they often go to research hospitals where the care will be provided for free, and you will help to train new doctors, for example. And so Henrietta Lacks couldn't afford treatment for her cancer, so she went to Johns Hopkins and was treated. Now, during this process, they took a sample of her cervical cancer, plated it, and found that it grew into, the cells would continue to grow. But they never got permission to do this. And Henrietta Lacks sadly died of the cancer. And years later, her family found out that these cells, their mother's cells or their family members' cells, had been growing all this time. Okay? And that they were, in fact, one of the largest and most commonly used cell lines. And these were called HeLa cells. Like this. And that stands for Henrietta, H-E, and then Lacks. Years ago when I was an undergrad, the name Henrietta Lacks was unknown. We knew that they were samples that had been taken from a female patient, and they had all kinds of names. Some classes we learned them as Helen Lang was one, but we now know that these were from Henrietta Lacks. And so there's all kinds of questions, ethical questions surrounding this cell line. It's been extremely important for the scientific and medical community. The polio vaccine was the first vaccine developed for polio, and it was developed in HeLa cells. The Johns Hopkins and the Gays, they didn't charge money for the cells, so these were freely distributed to researchers. So nobody was getting rich off of this, but on the other hand, they were used without permission. And it's not something that would happen now, hopefully. So now if you want to do anything with human tissue, there's a lot of paperwork, and we always seek for consent to that. And if we look, this is the most recent iteration. So Johns Hopkins has a number of different initiatives to sort of try to make amends for this, including symposia, different scholarships, things like that. But again, how do you correct a mistake of the past? And that's a theme that comes up quite often in science because, of course, a lot of things were done in a manner we would now consider unethical, even though the reasoning behind it was to improve people's lives. The approach wasn't necessarily correct. I think there's a question, yes? HeLa cells are still used, yeah. They're not used quite as commonly as they once were for a number of reasons. There's other cell lines, but yeah, there's still lots of HeLa cells. And we even use them, I think, in some of the undergrad labs in upper years. So, but yeah, still really commonly used. And one of the reasons they've been used for so long, and so many studies were done on these, that there's this huge wealth of information to go back to. And so if you're working with HeLa cells, you don't have to prove they do this, this, and this, because that's already been shown. Yeah, great question now. Yes?
If they were like transformed into malignant cells, then how were they used so many times as like normal cells?
Yeah, so if I kind of follow the question, you're saying since these are malignant transformed cells, and we keep using them for years and years and years, are the cells the same now as they were in the 50s? And they're not, actually. And we'll talk about that way later in the course, if that answers the question. It answers that question, but yes.
Like, well, if you were trying to like test how many cells, why
Oh,
would you use
okay. So the question is, why would we use these cells at all, transformed cells, to study humans? Because these are cells that are obviously already sick. What you would generally do is work with multiple cell lines simultaneously, because each cell line will have some quirk that's unique to the cancer. Yeah, good question. Okay, we are going to stop there. Have a great and a safe weekend. We'll see everyone next week.